Hello everyone and welcome to another recorded lecture of Anatomy and Physiology 1 online. Today we will discuss Chapter 3, which is on cellular form and function. As we mentioned in the first chapter, all organisms are composed of cells. And a cell is the smallest unit of life that can function independently. So here we can see some pictures of cells. So being composed of cells is one of the most basic characteristics of life. If you are not made of cells, you are not alive. Cells were not discovered until microscopes were invented, and that wasn't until the 1600s. So there are a lot of misleading theories until what we now know as cell theory was established. So once cell theory was established, we can start studying cells, and cytology is the study of cells. So the prefix cyto means cells. So like I said, before we had microscopes, we didn't really understand what life was and where living things came from. There was a very common idea of spontaneous generation. This is actually a real recipe, quote unquote. It was thought that if you wanted to make mice, if you wanted to make living mice, you could just take some dirty shirts, put some wheat in the pockets, and if you wait 21 days, you would get mice to form. Again, we know that sounds preposterous, but back in the day, they didn't really understand where mice come from. They didn't study the reproductive cycle of mice. And it wasn't until 1859 where Louis Pasteur did a very famous experiment that disproved spontaneous generation. You might have heard about this in bio already, but he took 3 flasks with meat in it. He left one unsealed, one sealed, and one covered with gauze. There was a common theory that flies came from meat, that living flies came from non-living meat. So this experiment, right, you can pause here and try to take a second to look at it. This experiment shows that flies were just attracted to the meat. The flies didn't come from the meat, right? They were getting tricked because they thought that the maggots came from the meat, but really the flies were just laying the eggs that turned into maggots on the meat. So this disproved the idea that non-living things can come out of living things, or vice versa, right? You can't get a living thing come from a non-living thing like meat. Things can't just generate spontaneously. So cell theory was first established in the 1800s after it became possible to study cells using microscopy. And the tenets of cell theory explain the commonalities of cells. It was established by a botanist called Schleiden, a zoologist Schwann, and a physician Virchow. So these 3 scientists together came up with cell theory, and it's very important for you to understand the tenets of cell theory. It says that all organisms are made of 1 or more cells. The cell is the fundamental unit of life, and that all cells come from pre-existing cells. That's cell theory. We now know a little bit more about cell theory. We know that all cells have the same basic chemical composition, they have the same biomolecules. We know all cells use energy, and we know that all cells contain DNA that is duplicated and passed on as cells divide. So that is cell theory. Despite cells having a lot in common, they come in lots of different shapes. So we'll encounter different shaped cells as we talk about different tissues in this course. So we'll see some discoidal shaped, some stellate shaped, some spherical, some fibrous, cuboidal, columnar, squamous, fusiform. So again, they come in lots of different shapes and sizes. Most cells are too small to see without a microscope. And if we were in class together, we would use light microscopes to view entire cells and tissues. An electron microscope is a lot more expensive, and those could be used to view parts of cells and viruses, which are much smaller and would require higher magnification. So just to show you this image, the range of the light microscope is how we can see most bacteria, most plant and animal cells. With the human eye, we can see frog eggs, which are single cells. Those are rather large, but we can't see anything a lot smaller than 200 micrometers. An electron microscope can view small molecules, proteins, viruses, and parts of cells. there are a few terms I want you to be familiar with when it comes to microscopy. Magnification is the ratio between the size of an image and its actual size. So an electron microscope can magnify an object hundreds of times more than a compound light microscope can. A light microscope can magnify things easily 1,000 times. But an electron microscope can go hundreds of thousands of times. Resolution is the minimum distance between two objects that allows them to be seen as two separate objects. So it's the ability to resolve to see two separate points. So a microscope with poor resolution might might let a student see one cell, but if I had better resolution, you might be able to resolve that it's actually two cells. And contrast is the difference in shading of an object compared to its background. So we need to have some objects stand out, so we need good contrast. Sometimes you can fluorescently label certain molecules to make them a different color so they stand out. And here you can see three different images. This one has high resolution, but very low contrast. You can't see from the background. This one has high contrast, you can see from the background, but low resolution. You can't see fine points. And here you have a nice picture with high contrast and high resolution. So one kind of electron microscopy is called transmission electron microscopy, and that's used to visualize internal cell structures. So it's almost giving you a cross-section. Right? It transmits electrons right through cells, and it gives you a cross-section of what the cell would look like. So here is a cross-section of a neutrophil, a white blood cell, and you can see all these organelles, these white blood, these lysosomes. You can see the nucleus. So it allows you to see very, very, very fine details. It can resolve objects 500,000 times better than the human eye. Another type of electron microscopy is called scanning electron microscopy, or SEM. And SEM reveals details on a cell's surface. So if you want a 3D image, very high magnification and high resolution, that's when you want to use SEM. You don't need to know how it works in detail, but you can spray the specimen with a thin coat of metal and then bombard it with electrons. And the electrons bounce off of the metal and can be detected by a camera. And this can give you an idea of what a 3D surface would look like. So SEM tells you the surface features, where TEM shows you the internal cross-section. And it said, finally, light microscopes have less magnifying powers than electron microscopes. They work by transmitting light through cells. And with electron microscopy, you can't view living cells because the electrons kill them. But with light microscopes, you can view living cells. And cells vary greatly in size. So most bacteria and archaea are only about 1 to 10 micrometers in diameter. A micrometer is a millionth of a meter. So most prokaryotes are 1 to 10 micrometers long. Frog eggs are the largest. Actually not the largest, because an ostrich egg, for example, is technically a single cell that we can see with our own eyes. But frog eggs are very large cells that are about 10 times larger than most plant and animal cells. Most plant and animal cells are about 10 to 15 micrometers in diameter. So I want to take a second and show you a quick activity. You can do this on your own by going to this link right now. So here is this activity that I just pulled up. And try to orient yourself. Imagine this is Times New Roman 12-point font. What I'm going to do is gradually zoom in so you can get a feel for how small we're talking here. So here's a coffee bean, here's a grain of rice, here's a sesame seed. So I'm going to slowly move in. This is the millimeter range. And here we have our first single cell, an amoeba, about half a millimeter. Then we see a paramecium. A human egg is rather large, and a skin cell, that's a good thing to remember, is about 30 micrometers. Then we have a red blood cell, it's about 8 micrometers, the X chromosome, a yeast cell, and now we get really small. Now we're on 1 micrometer. These are bacteria and organelles, like lysosomes and mitochondria. Then we get even smaller, we get to viruses. So viruses are on the nanometer, billionths of a meter. So viruses, and then we have very protein complexes like ribosomes and antibodies. Hemoglobin is a protein. Phospholipids, and we have biomolecules. And finally, the smallest is a carbon atom. And now I'm going to zoom out. So that's a carbon atom. Proteins, viruses, organelles, cells, bigger cells, and things we can see with our own eye. So we said like a skin cell is roughly 30 micrometers. So that's rather large, but a lot of our other cells are a lot smaller. But why? So why are cells so small? It turns out that cells need a large surface area of plasma membrane to adequately exchange materials. So we want a large surface area to volume ratio. Right? If I have just one big cell, that's not going to really be able to exchange as much materials in and out of the cell. But if I divide up the cubes into smaller and smaller parts, I have more surface area. Right? So I can transport stuff in and out of the cell a lot more efficiently. Regardless of cell type and size, all cells have the following, whether you're a bacteria or a human cell. You have DNA, ribosomes, a cell membrane, and cytoplasm, the stuff on the inside. All cells have chemical reactions taking place in order to sustain life. Right? So all cells do cell respiration and protein synthesis. Like I mentioned, one of the things all cells have in common are cell membranes, and cell membranes are made up of phospholipids. So cells are mostly water, and they live in watery environments, but they have a cell membrane that separates them from the external environment. We call this membrane a phospholipid bilayer because the membrane consists of 2 layers of phospholipids. Let's look at one phospholipid first. Each phospholipid has a hydrophilic head that has a phosphate group in it. That's why it's called a phospholipid. And then it has 2 hydrophobic fatty acid tails. Right? That's the hydrophobic portion. It's the fatty acid. It's a bilayer because phospholipids arrange themselves in 2 separate layers where the fatty acid tails face each other and the hydrophilic phosphate head faces the outer watery environment and the inner watery environment. So there's water on the inside of the cell and on the outside of the cell, and the fatty acid tails of the phospholipids are hidden. They're buried in between the hydrophilic heads. So make sure this makes sense to you, right? This is the organization of the phospholipid bilayer. So the cell membrane is selectively permeable. This means some things can get in and out, but not everything. So it allows oxygen and nutrients in, it allows wastes out. And the cell membrane is very important to control what can get in and out. So small hydrophobic molecules, so small nonpolar molecules easily get in and out of the cell. No problem. So lipids and small nonpolar molecules get in and out very quickly. Hydrophilic molecules need help from membrane proteins in order to enter the cell. So if you're hydrophilic, if you're a polar molecule, you need help from membrane proteins to enter the cell. Again, this is simplifying it a little bit because small polar molecules might be able to kind of sneak in through the phospholipids, but for the most part, like sugar, like glucose, is a polar molecule that can definitely not get inside of the cell. Sodium chloride, salt, like an ion, cannot get inside of the cell. It needs help from special membrane proteins. I want to illustrate this by an analogy. So I said that the hydrophilic heads are on the outside, and the hydrophobic fatty acid tails are on the inside. It's almost like a bread and butter sandwich, where the butter on the inside represents this hydrophobic portion. So suppose I wanted to get a hydrophilic substance, something watery, from outside of the cell, inside of the cell. So like, here's my watery dye, my hydrophilic dye. If I were to put it through the membrane, unfortunately, it would be blocked by the hydrophobic portion, right? This part represents the fatty acid tails. And that would not be able, so the dye, which is watery, cannot get through the fatty acid tails, the buttery portion. And that's a good thing, right? We want to have some protection. We only want information to get inside if it's needed. So membrane proteins can help transmit information from the outside of the cell inward, but they can also do a lot of different things. So different membrane proteins have many different functions. But a way you can think about some kinds of membrane proteins are like olives that are embedded within the phospholipid bilayer. Some olives without pits are representing channels. These represent membrane protein channels. This olive, right, or this membrane protein would allow something that's hydrophilic, like a watery dye, to go from outside of the cell into the cell, right? But again, it has to be a very special kind of a protein that's embedded in the membrane. This is called an integral membrane protein, and that can allow information from the outside in. So that's an example of like a transporter. But also we have lots of other functions of membrane proteins. Some membrane proteins are receptors that wait for something to bind to it on the outside, and then something on the inside will happen. So that's like this, a receptor. We said channels, right, allow things to go in and out. Other protein membrane proteins help cells attach to one another. So again, lots of different membrane protein functions. And membrane proteins are incredibly important in medicine because many of our drugs that we take actually target membrane proteins. So membrane proteins have a lot to do with physiology. So like I said, some of these membrane proteins are receptors. So what that means is that on the outside of cells, there are special membrane proteins. So this is a membrane protein that's stuck in the membrane that's waiting for a signal on the outside to bind to it. These signals are usually hydrophilic, right? So it can't just get inside of the cell and tell the cell to do something. It has to bind to this protein on the outside of the cell first. And when the molecule binds to the receptor, something will happen on the inside of the cell to tell the cell to do something. So these receptors, right, and the membrane itself act a lot like the brain. Let me explain. Right, what the membrane is doing, it's allowing certain things inside, but at the same time, it's blocking anything that's unnecessary. And that's what the brain does, right? The brain is a receiver, right? It's a processing tool. It keeps on interpreting information. It allows information in from the outside world so we can interpret it with our senses. But at the same time, our brain allows us to focus on one task at a time by excluding any irrelevant information. So just like when you're listening to this right now, you're not focusing on the cars outside or your screaming children because your brain is excluding unnecessary information, right? It's keeping you focused on the task at hand, hopefully. So the same thing is happening with the cell membrane. It's allowing information in, right? The cell needs to receive information from the outside world, but at the same time, it's excluding any unnecessary materials that should not be in the cell. So in that way, the membrane is like the brain, B-R-A-I-N, right? Like the brain of the cell, because it allows information in and excludes other information. So the membrane is incredibly important. That's kind of the sensory system of the cell, so to speak. And in reality, there's not just one receptor on a cell, right? So in response, so let's say we have a special calcium receptor. In response to calcium getting inside of a cell or binding to a receptor, you might have one protein. This is the membrane. all of this stuff is inside of the cell. So in response to, let's say, one calcium gets inside of the cell, that will activate one protein, which can activate another protein, which can activate another protein, which will activate another protein, which will activate another protein, which will then tell something inside of the nucleus to turn on different genes in response to whatever signal was on the outside. This whole thing is called signal transduction. And when you take an upper level biology course, you're going to have to memorize many of these pathways. So there's a lot going on inside of the cell beneath the membrane. But all this happens only if a certain signal binds to the membrane protein on the outside of the cell. Right? So when a ligand, also known as a chemical signal, binds to a receptor protein, so acetylcholine is a ligand or a chemical signal that can bind to a membrane protein receptor on cells, and something will trigger the cell to cause a response. Different cells have different response to the same signal. So for example, acetylcholine, if it attached to a receptor on a heart muscle cell, it will decrease the rate of contraction. And this is acetylcholine right here. You can see this is a polar molecule, can't just get inside of the cell. So it has to bind to a receptor first. If it bound to a receptor protein on a salivary gland cell, it actually tells the cell to secrete substances. If it binds to a receptor on a skeletal muscle cell, it actually increases contraction. So different cells respond to different signals differently. So now I want to give you a couple of examples of how these signals actually work. So sometimes you can have just one signal bind to a receptor, and that tells the cell something to do right away. That's like a first messenger system. This is called a second messenger system, because what we have is a first messenger happens, which increases the concentration of a second messenger inside of the cell. So they communicate within the cell receiving the chemical message, and an increase in the second messenger inside of the cell can then activate other enzymes. So let's talk about the example of the second messenger of cyclic AMP. Suppose a first messenger such as epinephrine, also known as adrenaline, will bind to its receptor on the plasma membrane. That's the first step. That will activate a special protein called a G protein, which is inside of the cell, which can then turn on a protein called adenylate cyclase. So again, in response to epinephrine binds to the receptor, that turns on a G protein inside of the cell, which activates adenylate cyclase. Adenylate cyclase can then convert ATP into cyclic AMP, CAMP, which is the second messenger. When there's a lot of cyclic AMP inside of the cell, it can then activate an enzyme called a kinase. Once a kinase is activated, that kinase can add phosphate groups to other enzymes, turning them on, and that can lead to a metabolic effect on the cell. So this is a run-through of a second messenger system that uses cyclic AMP. So let's give a specific example in the case of the beta-adrenergic receptor, which helps mediate the fight or flight response. So epinephrine is released from our adrenal glands when we're under threat, if we need to run away. It binds to special G protein coupled receptors. This is the protein receptor on the membrane of muscle cells. So again, our muscles need to be told we need to get more glucose into our bloodstream. So in the liver or the muscles, we said that we store glycogen, right? But if we need to run away from a threat, we need to turn that glycogen into glucose. So we have energy to run away from a scary object, right? Or we have enough energy to fight against the object. So what we need to do is, if we have an epinephrine, if we have that adrenaline rush, we need to get blood sugar increased. So we have energy to run or to fight. How do we do that, right? Epinephrine can't get inside of the cell and tell glycogen to break down into glucose. It doesn't work that way because epinephrine can't get inside of the cell. What happens is it has to bind to the receptor on the outside of the cell first. So it has a special receptor called the beta-adrenergic receptor, adrenergic for adrenaline. When epinephrine binds to the beta-adrenergic receptor, that will activate a G protein, which will turn on adenylate cyclase. Adenylate cyclase can convert ATP into cyclic AMP. That's the second messenger. When there's enough cyclic AMP, it can activate an enzyme called protein kinase A. You don't need to know that. But that kinase can then activate another kinase, which can activate another kinase called glycogen phosphorylase. Glycogen phosphorylase, when it's active, can finally break down glycogen into glucose that can be used for energy. So as a result of epinephrine being released into our bloodstream, we're then going to break down glycogen into glucose, so we're going to have more energy. We would never have had enough glucose in our blood if it weren't for epinephrine binding to our liver cells and telling our liver cells to make this enzyme activated. Right, so membrane proteins were the key to transmit this information from the outside in. So let's watch this video together to summarize the action of adrenaline on the beta-adrenergic receptor.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of cAMP from ATP. The cAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
Great, so you should be familiar with that. You don't have to memorize every step at this point. And so this is the breakdown that you can read. This is the minimum that you should know. So we spoke about membrane proteins, and on these membrane proteins, sometimes we find these chains of sugars, and these are really important. This is part of what's called the glycocalyx. The glycocalyx, and the glycocalyx is like a thin, fuzzy border outside of the cell membrane, mostly composed of carbohydrates. And it's chemically unique in every single person, but identical twins. So this is almost like an identification tag. It helps cells recognize each other, and it helps differentiate self cells from foreign cells. Inside of the membrane, there's a lot of cholesterol. So cholesterol, we said, is a type of lipid. And cholesterol keeps the membrane at the right level of fluidity. We don't want it to be too liquidy, too rigid, so cholesterol keeps it more like a jello, more like a gel. Right? And in the membrane, phospholipids are constantly moving and switching places. They're always diffusing in and out. So it's a very fluid, dynamic structure. It's not like it just stays in place. So the combination of phospholipids and proteins forms what we call a fluid mosaic. It's a mosaic because it has a lot of different pieces. And it's fluid because it's always moving. It's not a solid mosaic. So this is what we spoke about so far. We said the phospholipid bilayer has 2 layers of phospholipids. There's some cholesterol. We have membrane proteins that are integrated. You can have channels that allow things in and out. I didn't mention this yet, but some proteins can just be on the outside or the inside. Those are called peripheral membrane proteins. And here we have some lipids that have carbohydrates. This is a glycolipid, and this is a glycoprotein. This is a protein with carbohydrates attached. This is the fluid mosaic model. So pause here and see if you can answer this question. So the answer is D. So pause here and answer this question. The answer is A. Okay, now pause here and answer this question. The answer is A again. Now we'll discuss transport across a membrane. So what can easily cross the cell membrane? We said small, non-charged, non-polar molecules can easily cross the membrane. What is the membrane impermeable to? Well, water can sometimes kind of sneak in a little bit, but for the most part, we need special proteins called aquaporins to allow water in and out. Ions, charged molecules and polar molecules like sugars need special channel proteins or carrier proteins to allow them through. So, for example, glucose gets in through a carrier protein. Certain ions get in through a channel protein. Those are kinds of like the olives without pits. Right? So, large molecules, ions and polar molecules, and largely water require proteins to get inside of the cell. So how do substances travel across the membrane? It's by one of 2 mechanisms. Passive transport requires no expenditure of energy, so no ATP. And this is because random molecular motion of particles provides the necessary energy to move something passively. We'll talk about 3 kinds of passive transport: filtration, diffusion, and osmosis. There's also facilitated diffusion, which is similar to this. Finally, active transport does require ATP. We'll talk about active transport and vesicular transport, both requiring ATP. So filtration is pretty simple. Filtration is how particles are driven through membranes by physical pressure. So we can think about how water and small solutes can go through the cells that make up the capillary walls. So cells so water and solutes can be pushed out of the spaces between the capillary wall cells in order to get nutrients delivered into tissues. So again, just because of the force, because the heart, of course, is like a pump and it's pumping blood with a lot of pressure, that pressure itself is enough to force different molecules outside of capillaries. Simple diffusion, again, no energy is required. That's the net movement of molecules down a concentration gradient, going from high concentration to low concentration until equilibrium is reached. And equilibrium is when the solute concentration is uniform. So that means there's no more gradient. So this is a gradient. It shows more concentration on top, and then it goes less on the bottom. So if I were to spray Lysol in one corner of the room, there would be a gradient, right? There'd be a lot more of the Lysol particles in one corner. But over time, if I waited an hour, all those particles would diffuse, right? Diffusion. So there was equilibrium. The particles would be evenly spread throughout all 4 corners of the room. So over time, if there's a lot of molecules on one side of the membrane, what happens is they naturally go from high concentration to low concentration until there's an equilibrium on both sides of the membrane. Right, so this is an example of if you paste a crystal dye, those crystal dye molecules will diffuse into the water until there's an equal distribution of water and dye molecules. That just happens over time by Brownian motion. This happens in our bodies in gas exchange. So in our lungs, we have a very high concentration of oxygen gas in our alveoli, the little air sacs. And just by passive transport, by natural diffusion, the high concentration of oxygen, right, the oxygen will go from high concentration inside of the lungs to low concentration inside of the blood. And since oxygen is nonpolar, it can go right through the plasma membranes of the alveoli and the capillary walls, and that's how oxygen gets delivered to our blood. It goes from the high concentration of the lungs to where it's less concentrated in the blood. That happens passively. No ATP is needed. So several factors affect the rate of diffusion. So higher temperature can increase the motion of particles. The molecular weight, so the larger a molecule is, the slower it moves around. The steepness of the concentration gradient. So the more drastic the difference on both sides of the membrane, the faster the diffusion rate. If you have more membrane surface area, it will also speed up the rate of diffusion, right? You'll have more space to diffuse across. And then finally, which kind of makes sense, the more permeable a membrane is, the faster the rate of diffusion. Osmosis is diffusion that refers to the solvent water, the solvent movement rather than the solute. So it refers to water moving from one side to the other rather than the solute moving. And the net movement of water is always toward where there's high water is going to go to where there's less water. In other words, water is going to always move toward where there is more solute. So suppose you have a glass beaker with a divider. And this membrane divides the two halves, side A and side B, but the membrane is impermeable to solute. What that means is the solute is too big. It cannot cross from one side of the membrane to the other. But water is small enough to go from one side of the membrane to another. So what's going to happen after 30 minutes is that the water will go from high concentration on side A to low concentration on side B. So the water level will increase on side B, and that will increase the osmotic pressure. So osmosis describes how water will be drawn toward the high solute concentration, and that will increase the water level on side B. So solutions can be isotonic. That means the solute and water concentration is equal on both sides of the cell membrane. So if I were to put a cell in an isotonic solution, that means the salt in the solution is equal to the salt in the cell. If I were to put a cell in a hypotonic solution, hypo means less than. That means the concentration of solute in the solution is lower than the inside of the cell. In other words, that's saying that the cell is saltier than the outside of the solution. So think about this for a second. Will cells placed in a hypotonic solution swell? Will they grow? Or will they shrivel and shrink up? So pause here and answer. Right, so cells in a hypotonic solution will swell, right? Because the inside of the cell is saltier than the solution. So the water on the outside is going to want to balance all of that salt inside of the cell. So in a hypotonic solution, water will go inside of the cell, and the cell will swell up and possibly burst open. In an isotonic solution, water is entering the cell and leaving at an equal rate. So it's balancing the right shape. Hypertonic solutions mean that the solute concentration is higher in the solution than inside of the cell. And this would cause cells to shrivel up, right? This is also called crenation or shriveling, right? What's going to happen is, since there's so much more water on the inside of the cell, the outside is very salty, the water on the inside is going to want to leave to balance out the salty outside. So that's going to cause the cells to shrivel up. So think about like when you go into the salty water of the ocean, your cells kind of shrivel up because the water in your cells is trying to leave to balance out the salt in the ocean. So sometimes we need to use carrier proteins in order to transport materials across the membrane. And again, this is still passive transport. No energy is needed. So we're still going from high concentration to low concentration. But if the material we're transporting is polar, for example, right, it can't just cross the membrane by itself. So we need some help from a carrier protein. There are three different kinds of carrier proteins. You can have a uniport, which just transports one substance in one direction. You can have a symport, which carries two or more solutes simultaneously in the same direction. So two things go from one side to the other. An antiport can transport solutes in opposite directions. So as one thing goes in, another goes out. So we'll talk about two kinds of transport that utilize these carrier proteins. The first is called facilitated diffusion, where no energy is needed. You're going from high concentration to low concentration. Finally, we'll talk about active transport, which energy is required. That's when you're going from low concentration to high concentration. So facilitated transport is the use of carrier proteins to transport large or polar molecules across the membrane from high concentration to low concentration. So it's a variation of passive transport because no energy is needed. So suppose you have a lot of materials on the outside, a lot of solutes on the outside, but it's polar. It can't cross the membrane. One by one, it will bind to a carrier protein. The carrier protein will change its shape to allow it to be released to the other side. So this is how molecules will establish equilibrium. And no energy is required because we're going down the concentration gradient. All right, so good examples of this are like glucose and amino acids. Have to bind to carrier proteins, and then they can go down the concentration gradient by facilitated transport, also known as facilitated diffusion. So let's watch this video to summarize.
The plasma membrane is the gatekeeper of the cell, allowing certain substances in and out of the cell at certain times in certain amounts. Diffusion is a process in which substances move across a membrane from an area of high concentration to an area of lower concentration, or between areas of opposite electrical charges. This is called the electrochemical gradient. Small, non-charged particles, primarily gases such as oxygen and carbon dioxide, can diffuse through the plasma membrane by moving in between the phospholipids in the bilayer. However, the cell needs to control what enters and leaves, and so transport proteins aid in the selective movement of other molecules across the membrane without the input of energy. Through facilitated diffusion, larger molecules, polar molecules, and charged ions use channel proteins embedded in the bilayer. The transport of other substances requires a special carrier that will bind the substance on one side of the membrane, which triggers a conformation change in the protein carrier, causing it to release the substance on the other side.
So up until now, we were discussing how to transport molecules down the concentration gradient, going from high to low. All right, so it's pretty easy. When you go from high to low, that's passive. But what if we need to transport molecules against the concentration gradient? Right? What if we want to go from low concentration to high concentration? That is active transport, and for that we need energy in the form of ATP. Not only do we need ATP, but we still need special carrier proteins as well. All right, so ATP and a carrier protein are required to transport molecules from where they are lower to where they are already more abundant. That's active transport. And the major example of that is the sodium-potassium pump. You must know this example very well. It's going to come up many, many times when we talk about muscles and the nervous system. The sodium-potassium pump is an antiport that uses ATP while pumping sodium out of the cell and getting potassium inside of the cell. So this is very important. Each pump cycle consumes one ATP, so one ATP is used, and it pumps 3 sodiums out and gets 2 potassiums in. This is incredibly important because our cells need to have a potassium concentration higher in the inside and a sodium concentration lower on the inside relative to the outside. half of all the calories you eat every single day go to this one little protein. So it's pretty important. And every second, it can pump up to 10, it could do 10 cycles, up to 100 cycles per second. So again, we're using 10 to 100 ATPs per second to pump and to establish this proper electrochemical gradient. Right? Because again, it's really important to have this balance, or else our nerves won't be able to conduct electricity. And what's going to happen is sodium, since there's so much more outside, it's going to always want to try to leak back in, right, to establish equilibrium. But we have to use ATP to continually pump it out to make sure the inside has less sodium and more potassium than the outside. Cystic fibrosis is another example of what happens when active transport fails. Normally, we also have to pump chloride ions to the outside of the cell. We want very low chloride ion concentration in the cell, so we use ATP to pump chloride out of the cell. In cystic fibrosis, there's a mutation in that protein that doesn't permit active transport. So what happens is a lot of chlorine accumulates inside of the cell, and that attracts mucus on the outside of the cell. And mucus can accumulate in air ducts and the pancreas. And you can see over here, this is an X-ray of CF lungs. You can see how mucus is blocking the airways. So again, one protein that's defective can have drastic consequences on the whole body. So that's active transport. So far we were talking about processes that move one or a few molecules at a time. Vesicular transport moves large particles or multiple molecules at once in bubbles of vesicles. So like this is a vesicle. It's a bubble of membrane, like a little, it's a bubble of membrane. That's the best way to describe it. So a vesicle can carry groups of molecules in or out of the cell. And ATP is also needed. Right? In exocytosis, cells secrete contents out of the cell. So they release materials in exocytosis. In endocytosis, cells engulf in substances and internalize them in the cell. So exo means out, endo means in. We'll talk about exocytosis first. So exocytosis is the process of releasing molecules. So a vesicle from the inside of the cell that has a bunch of stuff inside comes to the outside of the cell and fuses with the membrane, and then all the stuff can be secreted to the outside. That's exocytosis. In endocytosis, we have the internalization of things, right? So the first type is called phagocytosis. That's cell eating. That's when you have a large particle being engulfed by the cell, and then it can be digested. This is how our white blood cells, our macrophages, can basically consume bacteria and viruses. They eat them by phagocytosis, a type of endocytosis. Here we have, macrophages are the good example. Pinocytosis is cell drinking. So pinocytosis is when cells take up small droplets of extracellular fluid that has some molecules in it. So pinocytosis occurs in all human cells, whereas phagocytosis only occurs in specialized cells. So pinocytosis takes in droplets of membranes in vesicles. And finally, receptor-mediated endocytosis is a more selective form of endocytosis that enables the cell to take in specific molecules from the outside because it binds to receptors on the membrane, which then become coated vesicles that are internalized by the cell. So this is a quick image that just shows phagocytosis, how it gets internalized by the cell. Then once it gets digested, it can then be endocytosed. It could be exocytosed. So this is endocytosis, getting inside of the cell. It can then be digested and then released by exocytosis once it's digested. In receptor-mediated endocytosis, there are some receptors that wait for their special molecule to bind to it. Once all the receptors bind to their molecules, they kind of cluster together and invaginate. They form a pit, a coated pit. That then forms a vesicle, and the pit separates from the plasma membrane, and then all of these molecules can then be delivered inside of the cell. So take a second and see, for passive transport, what direction are we going? High concentration to low or low to high? What is required? And give an example. So pause here and try to fill out this table right now. So please take the time to pause here and fill out the table for each of these 4 types of transport. All right, so here's the answers. And I'll talk a little bit more about what this means. So now we are ready to discuss the inner life of the cell. So as we're about to discuss the function of cells and how the different structures within a cell all work together, it's interesting to kind of compare a cell as a mini human. So what are some major functions of the body? I want you to pause here and just come up with a list of what all bodies do. So I'll show you a list of something that I came up with. Right, so we eat, we need to move, we need energy, we digest, we grow, we die, reproduce, evolve. Right? These are what human organisms do. And for every activity, for every function that's required of a body, the same happens on a smaller scale within the cell. So yeah, eukaryotic cells have organelles, just like humans have organs, right? And it's interesting and important to try to come up with analogies, right? So for every organ of the human body, there's like a comparison of an organelle inside of a cell that does an analogous function. So let's start by talking about the cytoplasm. The cytoplasm fills the interior of the cell between the nucleus and the plasma membrane. So the cytoplasm is mostly everything inside of the cell, and it's composed of cytosol, which is the fluid portion. The cytosol is a gel-like fluid that has salts and small proteins dissolved in it. It's mostly water. So the cytosol is the blue part. The rest of the cytoplasm consists of organelles. And the organelles perform specific functions. So organelles in the cell have specific functions, right? Organelles literally mean little organs. And they're surrounded by the cytosol, which is the fluid. So together, organelles and the cytosol make up the cytoplasm. And organelles have different shapes and structures, which of course means they have different functions. Let's start with the nucleus. The nucleus is the largest organelle, and the presence of a nucleus indicates that the cell is a eukaryote. So humans are all eukaryotes. We're only going to discuss eukaryotes in anatomy and physiology, so all cells we'll discuss have a nucleus. Bacteria, which we discuss more in biology, are prokaryotes and do not have a nucleus. The nuclear envelope surrounds the nucleus and separates it from the cytoplasm, and has nuclear pores that allows the movement of materials between the nucleus and cytoplasm. The nucleus stores chromosomes that are made up of DNA and proteins. Again, the nucleus of a cell is different than a nucleus of an atom. The nucleus of an atom had protons and neutrons. We're not talking about that here. The nucleus of a cell has chromosomes. Humans have 46 chromosomes per nucleus, right? 23 pairs of chromosomes. And the DNA of the chromosomes contains instructions that control the cell's function. And DNA is coiled into chromatin. So chromatin is the combination of DNA and protein. So here we have chromatin that's found inside of the nucleus. The nucleolus is a region within the nucleus where ribosomes are manufactured. You can see here's the nuclear envelope is a double membrane that has nuclear pores that allows things in and out. Ribosomes are cellular machines that make proteins. And ribosomes themselves are made up of a nucleic acid, ribonucleic acid, RNA. It's called ribosomal RNA specifically. And ribosomes are also made up of protein. So ribosomes are mostly rRNA, ribosomal RNA, and proteins. And ribosomes themselves make proteins. Ribosomes can be found floating in the cytoplasm or associated with the rough endoplasmic reticulum, which is right outside of the nucleus. And the endoplasmic reticulum is a system of flattened vesicles, continuous with the outer membrane of the nuclear envelope. And there's two varieties. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So if the endoplasmic reticulum, ER, has ribosomes on it, we call it the rough endoplasmic reticulum. And that's the site of protein synthesis. Proteins also get modified on the rough endoplasmic reticulum. The smooth endoplasmic reticulum is the site of lipid synthesis, and it does not have ribosomes. So the smooth ER is smooth, it does not have any ribosomes, and this is the site of lipid synthesis. It is also where calcium is stored in certain types of cells, like muscle cells. Muscle cells store calcium in a modified endoplasmic reticulum called the sarcoplasmic reticulum. And the endoplasmic reticula extend throughout the cell and is essential for protein transport throughout the cell and to outside of the cell. So pause here and answer this question. All right, so ribosomes are found. The answer is D. After the endoplasmic reticulum, proteins that were made on the ribosomes are enclosed in vesicles and are then sent to the Golgi complex, or the Golgi apparatus, as it's sometimes known. The structure of the Golgi apparatus is a collection of flattened membrane sacs called cisternae. And the function of the Golgi complex is to modify proteins that were made on the rough endoplasmic reticulum and to then direct them to their proper destination in the cell. So remember that the rough endoplasmic reticulum will make proteins, the smooth endoplasmic reticulum will make lipids. They get packaged into vesicles, but then the vesicles don't know where to go. So that's where we need the Golgi apparatus. So the Golgi apparatus can make carbohydrates and put some finishing touches on the proteins, and then it will tell those proteins where to be sent. So it sorts them and modifies them, and then it will say, okay, this belongs to the lysosome, let me send it to the lysosome. Or this belongs to the outside of the cell, let me send it there. So the Golgi is like a post person who looks at the different zip codes of mail and and sorts them to where they belong. That's what the Golgi is doing. It's looking at all these vesicles, all these envelopes of materials, and depending on different chemical signals on the vesicles, the Golgi will direct them to their proper destination. Right? So it sorts the vesicles for shipment. Those vesicles could be sent to different organelles. They can be exported from the cell by exocytosis, or they could be sent to the lysosome. Right? So after rough endoplasmic reticulum, smooth endoplasmic reticulum, they get sent to the Golgi apparatus, and then they can then be packaged in new vesicles and exocytosed or sent to a different part of the cell. Lysosomes contain enzymes that break down cellular components by hydrolysis. So a lysosome is like a packaged garbage disposal. And there are 50 different lysosomal enzymes that have been identified that break down proteins, nucleic acids, carbohydrates, and other phospholipids. So vesicles containing cell waste are sent to the lysosome, which then digest and dispose of those materials. And a very acidic environment is required, so a very low pH is critical for the lysosome to work. So what organ does that remind you of? It should remind you of the stomach, right, which is a very acidic environment that digests components into smaller parts. Tay-Sachs disease is a genetic disorder in which the enzyme hexoaminidase A is inactive. So If a lysosomal enzyme is inactive, that means it can't break down certain lipids. And what then will happen is lipids will accumulate in neurons, almost like a backed up garbage disposal, right? If you don't have this enzyme to break down the waste, all the waste will accumulate and grow and grow and grow. And when that happens in neurons, it can cause very, very devastating consequences. So Tay-Sachs is a genetic disorder that does not have very good prognosis because nerves can't function properly if they accumulate so much waste, and the lysosomes can't break that waste down. The lysosomes are essentially made by the Golgi because enzymes that are manufactured by the rough endoplasmic reticulum are packaged by the Golgi apparatus and made into new lysosomes. Also, debris from the outside of the cell can be fused, right? Can be endocytosed and then fused to lysosomes, so the lysosome can then digest any of that debris. So you can watch this video inside the book. So to summarize, we spoke about, this is called the endomembrane system, right? All the membranes inside of the cell. So proteins produced in the rough ER and lipids from the smooth ER are carried in vesicles to the Golgi. The Golgi modifies these products by adding sugar sometimes and then sorts them and packages them into vesicles. Some vesicles will carry the products from the Golgi to the cell membrane so they can leave. Others will go to different organelles or to the lysosome. And the lysosome is formed from sacs of hydrolytic enzymes that can digest micromolecules. So pause here and you can answer this question. The answer is A. So pause here and the answer is C. Now we could talk about peroxisomes. And peroxisomes also aid in cellular digestion, similar to lysosomes. But these are different. These are considered microbodies that kind of function separately from the lysosome. So they have separate enzymes. And what they do is they use molecular oxygen to oxidize organic molecules. So they detoxify molecules and metabolize lipids. Whereas lysosomes are not found in plants, peroxisomes are found in plant and animal cells. You can think about peroxisomes kind of like the liver, because just like the liver and the kidney kind of detoxify molecules and metabolize lipids, so do the peroxisomes. So kidney cells and liver cells have abundant peroxisomes. So they're really important for detoxifying alcohol and drugs, and for making sure that we're not getting poisoned. They're called peroxisomes because the reaction produces hydrogen peroxide, which is actually a toxic substance. So peroxisomes also have an enzyme called catalase, which breaks down that toxic peroxide into harmless water and oxygen. There's a disease called adrenoleukodystrophy, ALD, that's a result of defective peroxisomes. Normally peroxisomes have to break down any faulty matter, right? Any undesirable proteins, or fats rather. Sorry, peroxisomes will break down the fats. So if there are a defect in peroxisomes, you might have an accumulation of these fats, and that can cause damage to the white matter of the brain and the adrenal glands. And that can lead to mental impairments of ALD. So now we can discuss the mitochondria. So mitochondria generate cellular fuel by extracting energy from organic compounds, right? So energy isn't made by the mitochondria, it's more extracted by the mitochondria. And almost all eukaryotic cells have many, many mitochondria. Mitochondria have two membranes. They have an outer membrane that allows small molecules to pass through, and they have an inner membrane that is the site of ATP production, which is the main source of cellular energy. The inner membrane is folded into structures called cristae. And again, that helps increase surface area, so we can maximize ATP production in a tiny little organelle. The inside of the mitochondrial membrane is called the matrix. So inside the cristae is called the matrix. That's where ribosomes are found and enzymes that are required for ATP synthesis. Mitochondria also have their own DNA, which is interesting. So they have their own DNA that's found in the matrix of the cells as well, and they divide separately from the cell. So here we can see mitochondria. They have like a kidney bean shapes. They have these cristae, which increase surface area. The matrix is in the middle. And cellular respiration occurs in the mitochondria. That's how we extract energy from food. And the mitochondria requires oxygen to do cellular respiration most efficiently. So that's where C6H12O6, glucose, is oxidized into ATP, and then carbon dioxide, water, and heat are made as waste products. So overall, the mitochondria can extract about 38 ATP from one glucose molecule. Another organelle is called a proteasome. And the proteasome can help destroy proteins that are no longer needed. So proteins that are no longer needed are marked for destruction by a ubiquitin molecule. So here's a bad protein. It gets ubiquitinated, and then it gets automatically sent to the proteasome. So it almost gets like, it's a protein shredding tunnel, the proteasome. So as the protein goes through the proteasome, it gets chopped up into millions of pieces, which can then be recycled. So the proteasome, the lysosome, and the peroxisome all kind of digest stuff. So finally, we will discuss the cytoskeleton and molecular motors. So the cytoskeleton is a crucial aspect of cell structure and function. So just like our own skeleton, right, the cell has to have a skeleton. So the cytoskeleton is not made of bones, but it's made of proteins. And there are many different functions that these proteins can have. So they can function to establish cell shape. So cells have different shapes because of the cytoskeleton. They also have mechanical strength. They're able to resist stress. Cells are able to move because of their cytoskeleton. And intracellular transport of organelles is also mediated by the cytoskeleton. What that means is, let's say a mitochondria needs to go from one place in the cell to another. The cytoskeleton can help make that happen. So what's the purpose of our skeleton? Right? What would happen if we didn't have skeletons? Right? We'd be just an unstructured blob that's unable to move. And that's what cells would be like if they didn't have a cytoskeleton. Right? Cells would just be blobs with no structure. They wouldn't be able to move. So the cytoskeleton has three major protein components that allow cells to move and have structure and withstand stress. So again, instead of bones in the skeleton, we use proteins. Three different kinds of proteins. The first are called microfilaments. Microfilaments are the thinnest filaments, and they're made up of actin proteins. Intermediate filaments are intermediate in diameter, and they're composed of ropes, like protein ropes that are wrapped around each other. And finally, microtubules are hollow tubes. They're the largest in diameter, and they're composed of tubulin proteins. So this is just another schematic. You can see here's actin. It gives lots of cells their specific structure. Here we have intermediate filaments. This is a major component of feathers and hair and structural components. And then here we have microtubules. And it's showing a chameleon here because this chameleon utilizes microtubules to deposit different colored pigments on its external surface. So let's talk about each of the three types of cytoskeletal proteins. The first we'll talk about are intermediate filaments. So intermediate filaments are major for support and structure. So when you think about support and structure, that's intermediate filaments. So intermediate filaments are what help join adjacent cells together. They help keep cells intact, especially with epithelial cells, which have to be in one sheet, all attached to each other. So intermediate filaments helps support between cells. Also, like this porcupine spikes, or the nails, or feathers, or hair, these are all very rich in intermediate filaments. So like hair and nails are very structurally strong, and that's because they're abundant in intermediate filaments. One type of intermediate filament is a keratin. So you know keratin is a structural protein. And intermediate filaments are kind of pull and peel Twizzlers because they're a bundle of 8 different ropes that are wrapped around each other over and over again, and that's what gives them so much strength. So we said we need intermediate filaments for several different reasons. So suppose I had a sheet of epithelial cells with intermediate filaments and then without intermediate filaments. Suppose I want to stretch each of these sheets of cells. What would happen over here and what would happen over here? So pause right now and tell me what you think you'd expect to see. Right? If you have intermediate filaments linking adjacent cells together, then the cells will remain intact even when they're stretched. However, without intermediate filaments, if I were to stretch that sheet of cells, all the cells would rupture. And unfortunately, that's what's seen in epidermolysis bullosa simplex. So this is a hereditary skin disorder that's due to abnormally short keratins in skin tissue. And remember, keratins are intermediate filaments, and if they're too short, they won't be able to hold 2 skin cells together. So if they can't hold skin cells together, that means that even in response to very delicate movement, the skin cells can separate and form blisters. So this is a blister, it's a space where these cells should be attached to each other. This can cause the rupturing and blisters. So it's a very painful disorder to have. The next category of cytoskeletal proteins are microfilaments made of actin. Actin is really important for cell strength, but also for cell movement. So for cell locomotion, it's very important. Actin is also important in cell division. I'll talk about that later on. And we'll talk about muscle contraction in a lot more detail, because actin is very heavily involved in muscle contraction. One other function that microfilaments have is to form microvilli, which are very small extensions. These are microvilli over here. These are small extensions of the membrane, about 1 to 2 micrometers in length, and this could increase the surface area of a cell by up to 40 times. So actin gives these protrusions their structure, so you can actually maximize the surface area of the membrane. So this could be important in like the intestines, where we want to maximize absorption of nutrients. So these microvilli make protrusions to allow for more surface area. We said that cell motility, meaning the movement of cells, is also done by actin. So pseudopods are called false feet, and they're protrusions of actin that propel motile cells. So what you're going to see is at this leading edge, that's over here, there's going to be tons of actin growing and growing and growing up here. And as actin grows and grows and grows, the cell will be moved upward. And down here, the actin will be destroyed. So what that's doing is it's basically pushing this cell upward by constantly growing on this side and depolymerizing on this side. So this is a single-celled amoeba, and it's polymerizing these actin in these false feet to explore new territory. So this is actin growing up here. It's pushing and pushing the cell forward. Back here, actin is shrinking. And here you can see the nucleus is actually being moved as well. That's not done by actin. That's done by something else. But again, that's how cells move, by actin polymerization and depolymerization. So pause here and answer this question. The answer is C. They do not help cells move around, right? They do the opposite. Finally, we'll talk about microtubules. And microtubules are the tracks on which cellular materials are transported. Microtubules also provide support for the cells, just like actin and intermediate filaments. But microtubules are really important for transport. Microtubules are made up of tubulin dimers. So there's an alpha tubulin and beta tubulin that are attached to each other, and it goes alpha, beta, alpha, beta, alpha, beta. They kind of line up in protofilaments. And one microtubule is made up of 13 of these protofilaments with the hollow center called the lumen. So microtubules are tubes, made of tubulin. And in eukaryotes, the microtubules originate from the microtubule organizing center, which in animal cells is called the centrosome. So the centrosome is right outside of the nucleus, and within the centrosome there are two centrioles, and microtubules grow from the centrosome. The two centrioles are located in the centrosome of animal cells, and those help form the microtubules. And centrioles become very important in cell division because those give rise to cilia and flagella as well. We'll talk about that in a couple of slides. So centrioles help form microtubules. Anything regarding microtubules, right, relies on centrioles. So cilia and flagella rely on centrioles. So we said that microtubules are made from tubulin, but there happens to be a direction. So all microtubules are attached to the centrosome. Here's the centrosome. All microtubules are attached by their minus end. And again, this is kind of just arbitrary. The minus end denotes where the alpha subunit is closest to. The plus end is the free end where the beta subunit is closest to. So microtubules have their minus end attached to the centrosome with their plus end toward the periphery of the cell membrane. Microtubules grow at the plus end by addition of new tubulin subunits, and they shrink by the release of tubulin subunits. So they grow or shrink at the plus end only. So where are microtubules assembled? Answer is C. So microtubules are not just like train tracks that stay still. They're very dynamic. So they're always moving around. And this is called dynamic instability. Here's the centrosome, and these are microtubules labeled that are growing and shrinking. So this is called dynamic instability. And this allows microtubules to go to every corner of the cell. So again, it's not just like train tracks. It's like a train track system that keeps on moving. So we need to transport materials on these train tracks. But how? How do materials get transported throughout the cell? There are two major groups of motor proteins that can travel on microtubules. So motor proteins are proteins that actually move. Kinesins move toward the plus end of microtubules, and dyneins move toward the minus end of microtubules. So two different kinds of motor proteins. And so here's the minus end of a microtubule. This would be closer to the centrosome. Here's the plus end of a microtubule. And kinesins are motor proteins that can transport different cargo. This can be a vesicle maybe containing materials. This can maybe be in mitochondria. This can be a different organelle. But these motor proteins physically walk on microtubules, carrying these cellular cargoes to where they need to be. But remember, kinesin can only move in one direction, can only go toward the plus end, right? Dynein can only go toward the minus end. Right? And remember, the microtubules are not permanent structures. They are very, very dynamic. So this is how organelles get transported within the cell. This is how neurons signal and the brain's function, right? Neurotransmitters have to be released at the end of a cell. So vesicles of neurotransmitters can be carried along by kinesins on microtubules. So then the vesicle can fuse with the plasma membrane and be secreted from the cell. So you might recognize this picture from one of the first videos I asked you to watch, which was the inner life of the cell. This is showing a microtubule, and here this is a kinesin motor protein that's carrying a huge vesicle of stuff, and this is drawn to scale. So what's happening is this kinesin motor protein is carrying a vesicle and is taking step by step, walking on this microtubule, this train track, as I've been calling it. So step by step, this kinesin motor protein is bringing along this vesicle so it can be transported to a different end of the cell. And if we were to look up close, we would see that kinesin requires ATP, very much so. So with one ATP, I'm going to show you this one cycle. When one ATP gets hydrolyzed, this head over here is now activated. So what's going to happen if it's activated, that means it can take one step. So it's going to go over and take one step and then bind to the tubulin. And now this one had another ATP. That's going to use the ATP, and now this can take one step. And now this will take one step over that way. So again, you can imagine that there's a lot of ATP hydrolysis because you need kinesins to walk very far on microtubules. Microtubules are also responsible for cell movement. So cilia and flagella are extensions of the cell membrane that contain microtubules. And motor proteins, specifically dyneins, power the motions of cilia and flagella using energy from ATP. So flagella have a more propeller-like motion, and we only see flagella in sperm, an animal in the human body. Cilia, we see a lot more. On certain kinds of epithelium, we see cilia, which are little hairs, and they beat back and forth. Right, so they're short hair-like projections, and they help move substances along cell surfaces. So the digestive tract needs to move along substances. The respiratory system, right, we need to move down any dust or pathogens away from our mucosa. Flagella, right, are responsible for cell motility, and again, these are filled with microtubules. So sperm can move because of microtubules. Right? So within the sperm, within cilia and flagella, microtubules slide against each other with the help of the motor protein dynein. And ATP is required, of course, but what this causes is bending of back and forth. So this ATP hydrolysis can cause this flagellum to beat back and forth. Here's a quick video. It's hard. I just want to show you. Here you can see two flagellas. This is a chlamydomonas that's kind of walking around. And then here, let me show you some cilia. So here you can see some cilia. It's very hard to see, but this paramecium is beating its cilia to move through this liquid. All right, so you can see how those little hairs are beating back and forth. So that is the end of this chapter. So hopefully you have a better understanding of what's going on inside of the cell and how cells transport materials. And we'll talk a little bit more next time about molecular biology of the cell. So until next time, have a great day.